Hey guys, today we're going to talk about DNA replication. We already went over the fact that DNA replicates during the S part of interphase, but we're going to look more in detail about how that process actually works. Okay, so DNA replication. We're going to be talking about the different parts of DNA replication or the different kind of stages. We're going to talk about some of the different molecules that are involved in the process. And then we're going to briefly look at the difference between DNA and RNA and the different types of RNA. So DNA replication has to happen before any type of cell division can occur, whether that's meiosis or mitosis. In order for that division to happen, there has to be 2 copies of all of the DNA. So essentially that's how you go from like a single-stranded chromosome to a double-stranded chromosome that can then get pulled apart during the division process. So yeah, when DNA replicates, that is when we make the sister chromatid to our single-stranded DNA. So in order for DNA to replicate, it's going to go from that kind of ladder shape that we talked about yesterday into 2 kind of separate half ladders. So that's the complementary base pairs that make up like the runs of the ladders are what's called unzipped. So they're essentially just separated, those hydrogen bonds separate, and then the old strands become a template that we build the new strands on. And so that's how we get our new DNA molecule. So this is how it's going to happen. It's called semi-conservative because each of the 2 new DNA molecules are going to be half old and half new DNA molecule. So this kind of purple ladder is like the original DNA molecule, and then again, those hydrogen bonds are broken, so we have the 2 half ladders that are purple, and then each of those half purple ladders become the template to build the new half of the DNA molecule on the new strand. So there are 3 different kind of stages to this process of DNA replication in eukaryotes, which again are organisms like us that have nuclei. The first stage is called initiation. This is when the DNA is just kind of prepared for the new strand to be built. Then elongation is the actual building of the new strand. And then lastly, termination is essentially just kind of finalizing those new DNA molecules. This process of DNA replication can occur at multiple different parts along the DNA molecule at once, which just kind of speeds up the process. It can often be initiated at hundreds or thousands of different replication origins at once. So we'll look at initiation first. And it is important to memorize all of these different molecules that I'm going to be talking about and what their jobs are. So the first is gyrase, and it just goes and kind of unwinds the DNA molecule. Remember, DNA is like twisted up in that helix, so it's just going and kind of untwisting it to relieve some of the tension. Then helicase comes along, and helicase is the one that actually cuts the 2 strands of the DNA molecule apart. It's separating the complementary base pairs by breaking the hydrogen bonds between them. And the point where it goes from like 2 strands that are attached to 2 separate strands is called the replication origin or the replication fork. There are then some proteins that will bind to those 2 separated DNA strands so that they don't just like magnet together again. The DNA molecules will like hold them apart from each other. Then you have a molecule called primase, which attaches RNA primers to the 5' end of the DNA strand. The RNA primer is just kind of like a jumping off point for the building of the new DNA strand. The molecule that builds the new DNA strand is called DNA polymerase 3. And DNA polymerase 3 can't just attach onto a DNA molecule. It has to first attach onto one of these primers, and then it can start building from there. So the primase just attaches one of these primers so that the building process can start. So that gets us into the elongation portion, which is the actual building of this new DNA molecule. Again, that's done by DNA polymerase 3, which will start at the RNA primer and then just add nucleotides one at a time along the parent strand. So if the parent strand has like an A nitrogenous base sticking out, the complementary nitrogenous base to that is a T. So the DNA polymerase will take a nucleotide, the sugar, phosphate, and nitrogenous base that has a T for a nitrogenous base and bind it to the A on the original strand. And those will just hydrogen bond together. The DNA polymerase 3 can only move in the 5' to 3' direction. That's the direction that it's building the new strand, which means that it's moving along the parent strand in the 3' to 5' direction. And I am going to have a picture to go along with all this, so hopefully it'll make more sense. Because DNA polymerase 3 can only build in one direction, when it's building the DNA along the two different DNA strands, it's going to be moving consistently in the 5' to 3' direction along one of those strands, and that's called the leading strand. Essentially, the DNA polymerase 3 is moving towards the replication fork on the leading strand. The lagging strand is built more slowly. It lags behind the other strand because it's built in little tiny sections. And the reason for that is that it's being built in the opposite direction of the replication fork. So it like builds a little section and then backtracks, builds a little section, backtracks, builds a little section. You'll see all that when we get to the diagram. So these kind of dark blue are the original strands of the DNA molecule. They have been separated by the helicase. And then this kind of blue square is the DNA polymerase 3. It's building the new strand, which is this light blue color, in the 5' to 3' direction towards the replication fork, which would be right here. So that would be the leading strand up top. And then on the bottom, originally this DNA wouldn't have been open this wide. It maybe only got open like this far first. And so on the lagging strand, the DNA polymerase would have first done like this little section. And then as the helicase opened up more of the DNA molecule, the DNA polymerase would backtrack and start here and build forwards. And then as more got opened up, it would backtrack and then move forwards again. So that's why it's built in these little tiny chunks. Those little chunks of the lagging strand are called Okazaki fragments. And they do have to be bonded together afterwards. And that bonding together is done by something called ligase, which just creates bonds along the backbone between the different Okazaki fragments. Okay, I do like this kind of top picture here, this purple and blue one. Again, you've got the purple, which is the original DNA molecule. The helicase is unzipping it or breaking apart the hydrogen bonds. And then the leading strand is going to be built continuously towards the replication fork in the 5' to 3' direction. The lagging strand, again, has to be built in little chunks because it's being built as the DNA molecule is being unzipped. So when it's only unzipped this far, the building of that new strand would start like here and be built towards the 3' direction. And then it would backtrack 5' to 3', backtrack again, 5' to 3'. And then you get all of those little chunks of Okazaki fragment that the ligase will attach afterwards. And then in order to kind of finish off your new DNA molecules, there is something called DNA polymerase 1 that's going to cut off the RNA primers. So if we go back for a second, you can see this little red part right here. That is the RNA primer. That needs to be there in order to start building the new light blue strand. This DNA polymerase III molecule cannot start building without first having that primer to latch onto and then build from there. So at this end right here, we're going to have a primer, and then at the other end of the molecule, you'll have a primer over here as well. So both of those get cut off by the DNA polymerase III, and then the parent part of the DNA molecules that the primer was attached to just fall off because they're just like hanging single-stranded there, and then they just like fall off. So that would be like this part. So this is the primer. The purple on the other side of that red primer would just fall off because it can't just hang out single-stranded like that. So DNA molecules do constantly get shorter and shorter every time they replicate because you're cutting off the primers on either end, and then the other part falls off. So every time DNA replicates, it gets shorter and shorter. Now, obviously, you don't want to be losing important genetic information every time you cut the ends off a DNA molecule. So there is something called telomers that are at the end of DNA molecules. And telomers are just repetitive nucleotides that don't actually mean anything. They're just gibberish. They don't code for any proteins. Usually they're just TTA, GGG, TTA, GGG, TTA, GGG. And then in the middle of the DNA molecule is like the important stuff that you don't want to lose. So those telomers kind of act as a buffer zone that, again, every time the DNA replicates, you're losing some of that buffer zone, but you're not going to get to the important DNA information in the middle. So yeah, once the telomers get too short, then the cells will die. There is an enzyme called telomerase that can lengthen the telomeres, kind of like build on the end of the telomeres, that can extend a cell's life. And we find that enzyme in bone marrow cells and germ cells, so like sperm and egg, as well as cancerous cells. So yeah, there are like some researchers who are looking into like extending human lifespans by using telomerase to lengthen telomeres, but thus far it causes cancerous tumors. So still a way to go on that technology. Anyway, once the telomeres are cut off, then the DNA molecule will retwist into that helical shape, and then that's it for the DNA replication. There are, however, sometimes mistakes made during DNA replication. This is how we get some of our mutations, like our gene mutations. So you might have a nucleotide that is put in the wrong spot or doesn't get put with its like complementary base pair. And so both DNA polymerase I, that's the one that cuts off the telomeres, and DNA polymerase III, that's the one that builds the new strand, both of those will kind of go over the new strands of DNA to see if there's any mistakes and to replace any of those misplaced nucleotides. But not all the mistakes are caught and repaired, and if they're not caught and fixed, that's how we get new mutations. If the mutation happens in a germ cell, so a sperm or egg, that's how mutations then get passed on to offspring and end up in like the next generation. Mutations can also lead to cancer, and the rate of mutations will increase if the person is exposed to mutagens, which are things that cause mutations, like chemicals or radiation and stuff like that. So that's how DNA replication works in eukaryotes. It works a bit differently in prokaryotes. Firstly, prokaryotes don't have a nucleus, so this isn't happening in the nucleus for them. And secondly, DNA molecules in prokaryotes are circular instead of linear. So the enzymes will bind to the replication origin and separate the two strands, but this time, instead of having one replication fork traveling in one direction, there's two replication forks traveling in opposite directions, and you form what's called the replication bubble. Once again, the helicase is going to be separating the nitrogenous bases by breaking their hydrogen bonds. But this time, DNA polymerase will be adding in nucleotides, but again, in both directions. There are no telomeres this time because nothing has to get cut off because there's not like a straight line. Again, it's a circle. So this is kind of what it looks like. So yeah, you have the replication fork, which would be one here and one here. This whole area is the replication bubble. And so you'd have a new strand being built this way along, say, the outside, and a new strand being built this way along, say, the inside, or vice versa. And then you end up with 2 circular strands when you're done. Okay, the last thing I briefly want to talk about is the difference between DNA and RNA. So DNA we've talked a lot about. It is made of the deoxyribose sugar, which is where the D in DNA comes from. It is double-stranded. It's fairly long. It has the nitrogenous bases A, T, C, and G, and there's only one type. RNA has a different type of sugar. It's called the ribose sugar instead of deoxyribose sugar. It's only single-stranded instead of double-stranded. It's shorter than DNA, and it has one different nitrogenous base. So it has A, U, C, and G instead of A, T, C, and G. So it has what's called a uracil instead of the thymine. And there's 4 different types. So yeah, it still has the sugar phosphate backbone. It just has a different sugar in that sugar phosphate backbone. Again, it has uracil instead of thymine. So instead of having A always bonded to T like we do in DNA, the A will always be bonded to a U. And again, it is single-stranded. And these are the 4 types. There's the mRNA, which stands for messenger RNA, and the tRNA, which stands for transfer RNA. Both of those we're going to talk about in the next lesson about protein synthesis. There's also ribosomal RNA and small nuclear RNA, but we don't get into those types. Okay, so that's it for that. I do have a diagram of the DNA replication that I'm going to go over with you guys now, and then that'll be it. Okay, so this is my drawing of DNA replication. So here you can see that the DNA is still wrapped, like twisted up in that helical structure. So this is the gyrase. It's going to be moving in this direction, just untwisting the DNA molecule. And then following closely behind, we have the helicase. It's the one that's breaking these hydrogen bonds. So it's like cutting the bonds between our nitrogenous bases. And then where the helicase is, where we have this separation of nitrogenous bases happening, that's where the replication fork is. So the first thing that happens is that the gyrase starts untwisting. The helicase then starts cutting the 2 different strands apart. And then it'll just keep moving in this direction. Then we have down at this end here, the RNA primer is put on. Let me actually see if I can get a light on because you can't see my colors very well. Ugh. Honestly, I don't think that's any better, but that is the best I can do. So right at the end here, we have the RNA primer. It gets put on by the primase. And then this is the DNA polymerase 3. It would start here right after the RNA primer and just slowly build this leading strand towards the replication fork. Again, it's building in the 5' to 3' direction, which is along the 3' to 5' end of the parent strand. And then the lagging strand would be this new strand down here. It would start like right here. This would be its little primer. It would start right here and build in the 5' towards the 3' direction. And then as more of this strand opened up, the DNA polymerase would come back. It would latch onto this primer right here and again build towards the 3' direction. And then it would back up again and build in the 5' to 3' direction. That's why we get these little chunks of new DNA. Those are the Okazaki fragments. And the ligase will come in and it'll create a backbone right here to attach those little Okazaki fragments together. I do just have a couple notes. So again, the primers will be removed by DNA polymerase 1. Other than that, the new strand is always built in the 5' to 3' direction, which is 3' to 5' on the parent strand. So again, the new strand goes from 5' to 3', which is the opposite on the parent strand, because remember, DNA is always made of 2 anti-parallel strands. They go in opposite directions. And then at the end, DNA polymerase 1 and 3 will proofread for mistakes. Okay, so those are kind of the important things to know. Know your gyrase, your helicase, your DNA polymerase 1, your ligase, and your primase. Know what all of those do. Okay, let me know if you have any questions.